aren't we the lucky ones for the quality of the speakers we have? And that is going to be continued now with Professor Charles Ffrench-Constant. And I need to just read this to you, because I can't memorise it. Charles is Director of the University of Edinburgh Multiple Sclerosis Research Centre and Deputy Director of the Centre for Regenerative Medicine at the same university. He's also a Chair of Neurological Genetics at the University of Cambridge. I mean, an incredibly capable person, and he focuses specifically on the biology of myelin and the discovery of novel therapies in multiple sclerosis. So I'll now hand over to Professor Charles Ffrench-Constant, who will talk to us about stem cell development in the UK, and then we'll have some questions.
Doug, as ever, you're a hard act to follow. Now, where's the thing on this? Yeah. So, just a quick update on my career. I've now left Cambridge, in fact. I've been in Edinburgh now for 3 years, and for my sins, I'm now the director of the MRC Centre for Regenerative Medicine. And if the chairman's job is like herding sheep, my job is now like herding cats, in terms of the behaviour of some of my fellow academics. So what I want to do is very much cover the same ground as Doug. We discussed this beforehand, and there's a lot of commonality of the concepts and themes in our two talks, although the spin and the approach will be somewhat different. But I make no apology for this. I think whilst the sorts of things we're talking about now aren't in the clinic as we speak, stem cell therapies aren't in the clinic as we speak, this is coming down the track very quickly and will make a very big difference to all of you. And I think it's very important that everybody has a clear understanding of some of the very basic knowledge required to make sense of this very confusing area. Because most of what you read in the lay press is downright confusing and often just wrong. And so what I hope that both our talks will do is give you the primer that you need to better read what you see and be more critical about it. So, I'm going to talk about 3 things, as I make no apologies, again, you'll have heard some of this before. What types of stem cells are there? How can they be used? And then in the end, I want to very briefly acknowledge the subject I've been given to talk about, even if I largely ignore it, by discussing some of the ongoing developments in the UK by focusing entirely on the developments we're actually doing in Edinburgh. Now, you've heard about the 3 types of stem cell. This is my very simplified way of looking at human development. We start on the left as an egg, and we end up on the right as an adult. And we can get stem cells from pretty well every stage of that development. But the 2 basic sorts that you need to distinguish are the ones that come from the embryo, the embryonic stem cells, that can give rise to any cell type in the body. And then there's the tissue-specific stem cells that we get from adults that are more restricted and give rise to the cell types in a given tissue. Then in addition to that, there's a 3rd cell type you need to know about, which Doug's talked to you about, the induced pluripotent, or IPS cell. This is a technology that was discovered by Yamanaka in Japan, and he showed very beautifully that you could take pretty well any cell you liked and turn it back into a stem cell, which is going to completely revolutionize, as Doug said, what we can do with these cells, and I'll touch on that. Now, why do we have stem cells in our tissues? Well, one of the main reasons we have them is because they enable tissues to regenerate. And we all actually possess a significant regenerative capacity, as I'll show you, but some of our lower animal colleagues possess a remarkable regenerative capacity. And this is the example I like best. These are little 1 to 2 millimetre long organisms that live in the bottom of muddy ponds and rivers, called flatworms. And the experiment that I was lucky enough to do at school, which really started my whole interest in this area, was an experiment that was being done many, many years before by many biologists, where you simply take a very sharp razor blade and you cut the flatworm in half. And each half will turn into a new flatworm. And you can cut the flatworm up as many ways as you like and into as many pieces as you like, and each bit will turn into a flatworm. The really intelligent students in my class actually did the control experiment, which is they cut their finger at the same time with a very sharp razor blade. And they would have noticed that their finger didn't heal properly, whereas the flatworm did. And that actually tells you something very important about the difference in regenerative capacity. The reason these flatworms regenerate is because they've got stem cells present throughout their body. And those stem cells can all turn into a new flatworm. Now, you've heard that we have stem cells in tissues in our body. Indeed we do. And we do have them in the brain. And as Doug has told you very clearly, and I will show you again, actually they don't do a very good job of repair. So why do we have them? Well, there's some very interesting data suggesting the reason we have stem cells is because they might be important in learning and memory. It might be that you actually need these cells to generate new nerve cells to help us all learn and remember and change our behaviour. And the best example of this comes from this little bird that I put up on the screen. Now, I'm a Norwich City football fan. And it's because of you that I'm missing the home game against Scunthorpe. So thank you for that. My wife's going on her own and she's not happy. But anyway, so I obviously like canaries. And unlike Norwich City football fans, canaries sing beautifully. And not only that, is they learn a new song every year, which is definitely not like Norwich City football fans. And the reason they do this is because they need to find new mates. And female canaries choose their mate on the basis of how well they sing. Fortunately for us males, the rules are different for females. But for the canary, it's all about how you sing. And the way they do this is they have the neurons that are involved in generating song die every winter. And then in the spring, the stem cells in the canary brain activate and generate a whole new set of neurons, which enables the canary to learn a new song. And that's what stem cells do. And this is a nice example. They generate new neurons. So for the canary, it's very simple. Better stem cells equals better sex. Very important. And it's really, I think, a very nice example of why you have stem cells in your brain. So what can we use these stem cells for? Obviously, we have a slightly more sophisticated set of desires than the canary. Well, there are 4 things that we can use them for. And again, Doug has told you these, and again, I make no apology for the repetition. You need to understand the difference between these 4 applications in order to make sense of what's happening. We can use them for disease modelling using induced pluripotent cells. We can use them for drug discovery. We can use them for cell therapies. And we can use them for regenerative medicine. And now I'm going to give you examples of each of these. So let's talk first about disease modelling and drug discovery. This is where IPS cells come in. And this is work of my colleague Ian Wilmot in Edinburgh. That's the same Ian Wilmot who basically developed Dolly the sheep. So now, what Ian and a number of colleagues are doing is they're actually taking skin cells from patients with motor neuron disease who have genetic changes that cause that motor neuron disease, turning them back into stem cells, and then modelling the disease in the dish. This is very much what Doug already told you. This is a practical example of it. And in particular, this is a complicated slide, but I've put it up because it makes the key point that the protein we're interested in here is encoded by a gene TDP43, which is an important cause of motor neuron disease. And what you can do is you can basically take the cells from the patients that have got this faulty gene, turn them into the neurons or other cell types that are abnormal in the disease, and then study how they behave actually in a dish. And this is a really revolutionary way of looking at disease. So what Ian and his colleagues have done is they've taken skin cells, fibroblasts, from 4 patients, each with different genetic mutations. They've used these genes on your right here to actually make the IPS cells, and I'd be very happy to talk more about that if anyone's interested. And then they've modelled the disease, and in collaboration with colleagues both in the UK and in the States, they can now bring the very sophisticated battery of modern cell and molecular biology, some of the techniques of which I'll show you in a second, to bear on those cells. They can analyse the pattern of gene expression in those cells. They can see how the proteins that are abnormal in motor neuron disease actually behave, and particularly they can start to use the tens of thousands, tens of millions of cells that you can generate to try and discover new drugs. So this is a really important application of stem cells. And I think in terms of where the money is, this is going to be one of the big applications in the immediate future, because for the pharmaceutical companies, this is obviously a very important way of discovering new drugs. But the pharmaceutical companies can use this technology for another reason as well. When you're developing new drugs, a very large number of drugs will fail in the final stages of the clinical trials, the very expensive stages of the clinical trials, because unexpected toxic effects or unexpected side effects will appear. This is incredibly expensive. Now, the ability to generate very large numbers of human cells from different groups of people, all of whom are genetically different, or from people with different diseases, gives the companies another way of testing their drugs on a relevant cell before they actually go into humans. And so one good example is the ability to make liver cells. Now, as Doug told you, we can make all these many different cell types in a dish. And one of the cell types we can make is liver cells. Now, you're sitting here thinking, well, I don't give a stuff about my liver cells. It's the brain cells I want these people to be interested in. Well, that's true, but you need your liver cells because the liver cells are the primary cells that actually metabolize the drugs you're given, and a lot of the toxic effects of drugs actually happen on liver cells. So colleagues in Edinburgh, who I've listed here, are busy making liver cells, and you can see some in the bottom right in green, and you can make billions of these cells. And they behave just like liver cells. In fact, they behave so like liver cells that you can put them back in the liver and they will engraft. And what they're trying to do at the moment is they're making an artificial liver that you can use to keep patients alive when their own liver fails. So these are a very, very good source of cells for drug testing. Right, so that's the first two. What about cell therapies? Well, I was worried that I might have to spend time explaining the different uses of cell therapies, but Doug's done it for me very beautifully. There are two very different things going on with cell therapies. The first is that you can use cell therapies to actually replace damaged cells. So here you're giving a stem cell to actually replace the cell that's damaged. So if you're transplanting somebody with transverse myelitis in the application that Doug was talking about in the future, if you're transplanting someone with transverse myelitis, this would be where the cell actually generates an oligodendrocyte to form new myelin. But there's another very important application of stem cell transplants. As Doug has told you, stem cells have a very interesting property of they actually modulate the activity of the immune system, and they also produce factors, trophic factors we call them, that seem to keep other cells alive. So stem cell transplants can actually quite significantly depress the activity of the immune system. Now this is important because in many of the animal studies that have been done modelling disease, the overactivity of the immune system is an important reason why the animals get the symptoms they do. And the same is true in humans. And so it's perhaps not surprising that stem cell transplants into these animals have shown beneficial effects. But none of those, or very few of those studies, actually show that the stem cells replace the damaged cells. They're simply showing that the stem cells suppress the immune activity and protect the surviving, some of the cells that are still surviving. But it's on the basis of those animal studies that a lot of people are spending very large amounts of money to fly to unregulated clinics and get stem cell transplants. The stem cells they're being transplanted with will probably last in their body seconds or minutes, because there's no effort made to match genetically, and the cells will be rejected immediately. And I think a failure to understand the difference between these two applications and a failure to realise why the animal studies are giving results which actually might not be so relevant to humans is a very important driver behind this enormous problem of stem cell tourism that we currently have. Now, I want to talk about cell therapies just focusing on cell replacement, and I just want to give you a couple of nice examples that we're working on in Edinburgh, but are also being worked on in many other centres around the world. And one of them I've already talked about, which is the ability to generate liver cells. We can generate very large numbers of liver cells from a very wide range of stem cells, as you can see here on the left. And colleagues in Edinburgh have shown, as I said, that you can put these back into animal models of liver disease and show very clearly, and in light of what I've just said, this is very important, show very clearly that those stem cells will integrate and function in the environment of a damaged liver. Now, this is really important. Liver disease is a very common cause of death, and it's a cause of death that is increasing very rapidly. Contrary to what we would all think, liver disease is not only caused by alcohol. There's very large numbers of other causes of liver disease, biliary cirrhosis, primary biliary cirrhosis being a particularly worrying cause. There's a huge need for livers for transplants and nowhere near enough organs available for transplantation. So stem cell therapies for liver disease will be very important. Another nice application is blood. We have a very good blood donation service in the UK, as in many other countries, but nonetheless there is still more blood needed. And colleagues in Edinburgh and Glasgow have developed technologies for turning embryonic stem cells into red blood cells. And the science is on the left, but the image is on the right. And there you can see a nice little pellet of red blood cells that have been made directly from embryonic stem cells. Now, that's a millionth of the numbers of cells you would need for a blood transfusion, but nonetheless the proof of principle is very exciting. The thought that one could make limitless clean blood from stem cells at some stage in the future would have enormous impact on our ability to give safe blood throughout the world. Now, finally, regenerative medicine. Now, I'm using regenerative medicine to define very precisely here the ability of the body's own stem cells to repair the damage. So I'm distinguishing it from cell therapies, where the cells are being given to actually replace damaged cells. Here, you're actually using the body's own cells to do it. And I think it's just important to look, conventional medicines tend to focus on the cause of the damage. But many diseases actually result just as much from the failure of the subsequent repair as they do from the damage-causing process in the first place. So we desperately need drugs, like we hope lingo, that will actually, or anti-lingo, that we will actually hope stimulate the regenerative process. And we and many others are trying to develop such drugs, and I'll show you another example in just a second. But remember that this is to work alongside conventional medicines, not to replace them. As we've already heard, tissues will regenerate quite well in some cases. So let's talk about liver, for example. Liver has a remarkable regenerative capacity. So if we look at the example on the left to begin with, if you actually have a good night out and you damage a significant number of liver cells, they will disappear, but then the remaining liver cells around will simply replace them. However, if you really go for it, or you do drink much too much, or other causes of liver disease, what happens is the other cells are no longer able to repair and damage your liver. And now what happens is the stem cells, which are sitting in the liver next to tubes called the bile duct, activate, and they generate lots and lots of new cells, which turn into liver cells. And that's how you repair your liver normally. Now in the brain, that doesn't happen properly. So this is the brain of somebody with multiple sclerosis, a related disease, but distinct from transverse myelitis. And you can see here, these white lesions here in this blue area, which is the myelin in the middle of the brain, those have not repaired at all. But then there are other areas, these light blue areas here, in the same brain of somebody who's obviously died with this condition, where they have repaired. So why is it that those lesions have repaired okay, but the other ones haven't? But that tells us something quite important. It tells us that even in patients with long-standing disease, there is the capacity for repair. There are the cells there. For some reason, they just won't do the job. Now, my phraseology might not be as neat as Doug's, but the point is the same. These are the things on the log. I can't remember the exact word you used. But this is the same problem, basically. The cells are there, but they can't manage the repair process. And why is this important? Well, if we carry on using MS as an example, it's important because we know that if the axons, the nerve fibres, don't get repaired by new myelin, then they will actually degenerate. So the nerve fibres, even though they're not the primary target of the disease, degenerate because the repair process has failed. And that's a good example of why, in some diseases, it's a failure of repair that's just as important as what causes the damage in the first place. In MS, it's the failure of repair that causes chronic progressive disease. So we have to try and find ways of getting those stem cells to actually reactivate and do the business. Now, you've heard about one approach, anti-lingo, which looks very exciting. I believe it's in phase 2 clinical trials at the moment, and we're all keeping our fingers crossed. We were interested to see if we could find any new ones, and so a collaboration between Robin Franklin, who's my long-standing collaborator in Cambridge, we took the approach I'm going to show you in the next few slides to identify another potential way of enhancing remyelination. Just like Doug, we use animal models. These are absolutely vital for the research. We inject a small amount of a toxin into the cerebellum, a region of the brain involved in balance and often affected in MS. A small amount of the toxin into this area, and what we do is we create a little area of demyelination that we know will repair naturally on its own in a couple of weeks. And what we're trying to do here is we're trying to identify the factors that are normally increased when the repair is going on. So we know that in this particular case, the log, they don't just sit on the log, they complete the repair process. So we're trying to identify the factors associated with that completion, because if we could find out what they are, then the logic is we can stimulate them when they fail. So what we do is we take the brain of this animal at different stages of the repair process, and we use a laser beam to cut round the area of damage, and you can see the section here with this tiny little teardrop shape cut out with a laser beam. This is, less than a millimetre. And then we pop those little bits of section into a tube, and we analysed every single gene that's expressed in that little bit of tissue. And that generates a vast amount of data. And in order to actually make sense of that vast amount of data, the way we chose to do it was to do what's called a volcano plot. As we did this at about the time that most of us were stuck in various foreign countries because of the volcanoes in Iceland, it was particularly topical. But you can see here the volcano plot looks like ash being spewed out of this volcano down here. But what it actually shows you is the extent to which genes change their level of expression, either going down on the left or going up on the right, and the likelihood that that change is not due to chance going up the graph. So what you're interested in are the genes at the top left, which are the ones that are going down the most, and the top right, which are the ones that are going up the most. And we asked what genes change at exactly the time of that final stage of the repair process, which appears to get blocked. Only in this case, remember, we're studying a repair process that happens normally. And we found one particular gene right at the top on the right, RXR gamma, that had not been previously identified as a potential drug target for this condition. And so, to cut a long story short, what we then did was we took the same animal model, but now we injected a drug into the animal that activated RXR gamma during the repair process, and sure enough, we found that we could significantly accelerate the whole repair process. So that is another potential target. This is many steps behind lingo, because what we now have to do is we have to move on to the next step of drug development. But I show you the process because it gives you, I hope, an example of the logic that we use to identify new stem cell therapies to promote endogenous repair. So finally, what's happening in the UK? So as I said, I'm going to be very parochial here, and I'm just going to show you 3 or 4 slides of what we're doing in Edinburgh. But actually, I think what we're doing in Edinburgh is a very good example of what's happening in the UK. There's an enormous investment being made by government, by industry, and by academia in stem cell research in the UK, because we believe very strongly that this is going to be an incredibly important technology and therapeutic area in the future. And one of the things that you need to do to make this happen is you need to bring together the scientists who understand stem cells with the clinicians who understand the diseases and how to actually do clinical trials for which stem cells might be useful. And finally, you need to bring in a group of people who actually understand how to grow stem cells in conditions where they would be suitable for clinical use. That's not trivial. You can't just take a cell out of the lab and put it in a human. You have to grow it in an incredibly special environment that's completely sterile, where there's no contact with animal products and there's no risk of any animal diseases getting into the cells. It's called good manufacturing practice, or GMP, and it costs a lot of money. Now, what we've done in Scotland, and I think this is being done somewhere else, elsewhere, but this is unique at the moment, is we put all 3 of those groups into a single building, the Scottish Centre for Regenerative Medicine. This is a £56 million new build that will be finished in a few months' time. And it contains room for about 200 scientists, and the GMP facility and the animal facility and the clinical, not clinical facilities, I'll come on to those in a second, but the facilities for translational research that the clinicians need to bring all these groups together. And just to show you where it is, this is a view out of the aeroplane shortly before you land at Edinburgh Airport. And you can see here the hospital. This is the Edinburgh Royal Infirmary, which is a new build. Here's the medical school here, and here's the Queen's Medical Research Institute. And this building here is the Scottish Centre for Regenerative Medicine. And this is where we're all going to be based, in this building here. Now, there's a number of quite interesting features in this photograph, apart from the golf course. And there are many such golf courses in Edinburgh. What it shows you is what we're trying to do here is we're trying to bring together all of the different people involved into a single campus. So you can see here we've got the scientists and the GMP specialists working here next to a hospital here. Now in this building here, the Queen's Medical Research Institute, we have an imaging facility that is research only with CT and MRI scanning, which is absolutely critical for analysing the results of the clinical trials. We've got a very, very high quality MRI scanner. I don't think anybody's made love in it yet, but if they have, they'll certainly have needed to wear a hell of a lot more than socks with the winter that we've just got. But nonetheless, it's all here on this site. This is a few hundred yards, not, as in some other places in the UK, about 5 to 10 miles. And you need to bring all these people together. You also need to think very hard about the design of the building. And we've created a completely open plan research environment with core facilities in the middle. So all the graduate students and postdocs and other people working on these projects will get mixed up, because barriers, either physical or imaginary, are the enemy of collaborative science. And we've designed this building very specifically to get around that. But finally, just my last point, I've made the point already that this building won't have clinical facilities in it. Now, we do have a hospital here, But this hospital, like every other hospital in the land, is running at virtually 100% capacity in all its clinics. So if I went to the hospital manager and said, I want a research clinic for transverse myelitis or MS or MND, they'll just laugh at me. There is no capacity in outpatient clinics in UK hospitals any longer, and it's going to get worse, not better. So what you have to do is you have to have specialist clinical research facilities, which are only for patients going through clinical trials. And we were fortunate in Edinburgh in that recently the author J.K. Rowling gave us 10 million to build such a clinic for our MS work, but also for all our work on neuroregenerative disease. It's not just MS, it's for all neuroregeneration research. And this, I think, is going to be an absolutely critical component of the stem cells to the clinic, because we can't do that within the existing NHS structures, either in Edinburgh or anywhere else. We need these clinical research facilities. We will build a 7-room clinical research facility that will only be used for research. And I think this is the way that we and other UK centres need to go if we're going to realise the opportunities offered by stem cell research. So thanks very much.
Well, another fantastic talk, and I can only hope, Charles, that in return for giving up your afternoon, that the Canaries did the business, or have done the business, and won your game of football.
As long as Swansea don't win.
Well, exactly, absolutely.